Thanks to everyone at TOFS for asking me back. It's always a privilege to come and speak to you. My name is Charles Shaw-Smith. I'm a consultant in clinical genetics, which is definitely a niche specialty. What we want to do in clinical genetics is to find out what the cause of things is. And in esophageal atresia and TOF, we're not doing very well. So what I'm going to do is, and I'm only going to talk for about 15, 20 minutes, so don't worry. I'm going to review what we know about the causes. And I think people do want to know what the cause of things is. And I think really for this condition particularly, because it is so dramatic and sudden, it's like, why on earth did that happen? I think you deserve an explanation. And at the moment, you tend not to get one, which is unsatisfactory, to say the least. Just a slide about how the esophagus and trachea develop. A slide about what we know about, does it run in families? Which is always worth knowing about, especially if you're a geneticist. A bit about twins. And in fact, this talk is billed as the lost twin hypothesis, and I am going to talk about that, but it's really more of a general, there's only a couple of slides at the end about that. It's a research project that I'm involved in that some of you may have heard about. But really, I want to give more of a general overview as well. I think it's only fair to do that in future plans. So why does it happen? What do we know about the causes? This is a really rare one. So there are some medicines that you can take in pregnancy, but I think that's really, these are known about. One of the thyroid drugs, for example, methimazole, but that counts for hardly any. Maternal diabetes, maybe, if it's poorly controlled diabetes. Chromosome conditions, definitely. So Down syndrome, for example, has a much higher instance of esophageal atresia than the general population. And there are some really unusual single gene conditions, but they tend to be syndromic, which means that there's quite a lot of other things going on as well. So they're quite easy to spot. And in fact, the majority, well, it's actually about 50-50 of TOF, esophageal atresia, is non-syndromic. So that's the only thing that the child has. But of course, there's the VACTERL condition, which I'll say a little bit about, which is not non-syndromic because it's multiple organ systems. And then there are these single gene conditions, which are really small print. And although they're interesting in themselves, they don't tell us much about the generality of TOF and esophageal atresia. So you've got this ocean of unknown cause, which is really frustrating. And you can see that the percentages are, well, it's much more easy to appreciate from the pie chart. We just don't know what most of it is due to. So chromosome conditions, Down syndrome is the main one. There are in fact lots and lots of chromosome conditions, and every child with a syndromic version of TOF will have a chromosome test. And if you lose tiny bits of chromosomal material in various locations throughout the whole set of chromosomes, you may find other ones. But again, these are all really quite rare. The heritable conditions associated with OA-TOF do account for a very small proportion, but I think they're the best route into understanding in some ways, because you have a gene and you can look at what the gene is doing, and it's generally involved in the development of the esophagus and the trachea, unsurprisingly. But it's important to know about these conditions because they can run in families, obviously, and they tend to be syndromic rather than isolated. So if you are the parent of a child with this and you suspect there might be something else going on besides the esophageal atresia, do ask to be referred to see a clinical geneticist, which you're all entitled to do. There are genetic centres which cover every postcode in the UK, and it's quite a together service. So if you have concerns about that, or if you want to have another child and are worried about the chance that the same condition could recur in a future pregnancy, that's what we're there for. So ask your GP. All GPs know about us. They don't necessarily always refer to us, but they know about us. So these are the known conditions. I'm not going to talk about them in detail at all. Some of you may recognize the name on there. The FOXF1 one I'm proud of, because way back in about 2005, TOFS gave me some money to do research into this, and it led to a funding from the Wellcome Trust, and I was researching this and we found that. So that was my work in collaboration with some researchers in the States. But that condition, alveolar capillary dysplasia, that's a lethal lung condition. So those poor babies don't survive at all. So again, it's interesting but incredibly rare and not telling us much about most of what's going on with TOF. VACTERL association is really interesting and again very poorly understood. And it's more like esophageal atresia on its own, non-syndromic, than it is like the chromosome versions or the genetic versions, because you get these malformations, and I think that is the PC word to use now, although not everyone will like it, but we have to call it something, in more than one organ system. So if something is going on in more than one organ system, we tend to call that syndromic. And there's the acronym for VACTERL, so it can be the spine, the heart, the gastrointestinal system, the kidneys, the limbs. But in contrast with genetic or chromosomal conditions, where you quite often see delay in development, learning disability, and growth problems, you don't tend to see that with VACTERL. So it seems to be more in the isolated TOF camp rather than the genetic syndromic camp. And the other thing about VACTERL is recurrence, very unusual, like TOF, which I'll come on to. Just a word about the different flavors of OA TOF. From the perspective of a clinical geneticist, it's not helping us because you get all types and all different causes. So it's unhelpful, but just to recognize that those different types are there. And of course, they're extremely important for pediatric surgeons because they're the people who are going to fix it. Again, a brief mention of development just to make the point that this budding off of the trachea from the foregut, which becomes the esophagus, happens at a really surprisingly early stage in pregnancy, between 6 and 10 weeks from the first day of last menstrual period, or between about 4 and 8 weeks of embryonic life, when the embryo is really tiny. But it's critical to understand that anything that happens after that point cannot be the cause. So if you drove past a field that was being sprayed with some chemical at 15 weeks gestation, that wasn't the cause. Okay. And I know I mention that because things like that are things that people spend quite a lot of time dwelling on, but it's just to understand that point about development is important. So OA-TOF usually does not run in families. It tends to be a sporadic, isolated event without recurrence in a future pregnancy or in the offspring. And just that point about the surgery. So there was a geneticist at Great Ormond Street called Cyril Carter who did a very sensible thing, really. As soon as children with TOF started to survive and grow into adulthood and have their own children, he thought, well, let's see if it runs in families. Let's see if their children have it as well. And he published a paper about it. And in fact, so the vast majority, there's no recurrence, backing up what I said about the fact that it doesn't appear to be genetic. But there's one instance and one family, in fact, where there were, as you can see at the bottom, there were 3 affected individuals, 2 sisters and a son. And when you look at the description of what's going on there, it's clearly one of the heritable ones, called Feingold syndrome. I'm sure that's what it was. So if you exclude that family, then you're left with no recurrence, which I think is correct. So if we're talking about non-syndromic, isolated TOF, esophageal atresia, it'd be very, very unlucky. In fact, I think the chance of it recurring would be the chance of it happening the first time multiplied by the chance of it happening again, which is 1 in 3,000 times 1 in 3,000, which is about 1 in 9 million. So it could still happen, and it probably has happened, but it's very, very, very low chance. So just a bit about twins. Being a twin is, especially an identical twin, is a risk factor for a wide range of congenital malformations. So in the regular population, 1 to 2% are twins. And in infants with TOF, esophageal atresia, it's 2 or 3 times that. That's the first thing. Second thing, which is really interesting for a geneticist, is what's referred to as the twin concordance rate is very low, which means that usually just one of the twin pair has it and the other member of the twin pair does not. And in identical twins, if it's a genetic condition, they're both going to have it. It's obvious. So this always made me uneasy about being a geneticist studying TOF. It just didn't make sense. It caused me some sleepless nights. Third thing is, it's more about identical twins than non-identical twins. And people have got a kind of proxy for that by looking at whether or not the twins are like-sexed. And if it was, so non-identical twins are much commoner than identical twins in the general population. So you would expect, if it was mainly non-identical twins, that about 50%, this is a bit mathematical, sorry, about 50% would be of the same sex. But in fact, when you look at it, it's more like 70 or 80%. So that's not random. That's suggesting there's a heavy skewing towards identical twins. Make sense? So what can we conclude from this? Being an identical twin appears to be a risk factor for TOF, suggests a genetic cause, but TOF doesn't usually run in families, and identical twins are usually discordant. One of them has it and the other one doesn't. So there must be something else going on. This is getting into my lost twin hypothesis now. So we know twinning is a risk factor for TOF. What about a twin that was present at the beginning of pregnancy but subsequently lost, so was not evident? And there is some evidence that early loss of a twin is associated with an increased risk of a variety of malformations, but not specifically TOF. So I've been scratching my head about this for quite a few years now, and I've tried various ways of investigating it. I started off, and Sue Lewis-Jones helped me with this, doing a survey of the TOFS membership to try and investigate whether there was evidence of a lost twin, which was interesting, but didn't provide me with the evidence that I needed. I then tried to turn that into a case series for publication, but that foundered somewhat. I then started a collaboration with a fetal medicine specialist, or tried to, who had access to a really detailed registry in Denmark of all births, where they've got all the records going right back. And I thought, if I could just go into that database and see if there was an early twin that was lost, and then look at how many of them had TOF, that would be quite useful. But it came to nothing. There've been a lot of false dawns with this, and I'm still not through with them. So what I needed was a way, and this was a real light bulb moment for me. What I needed, I'm just looking at the time. How am I doing? All right. What I needed was some test that would tell me, if you put a child in front of me with TOF, was that child at some point in his or her life a twin, going right back, that was not diagnosed? Sometimes the twin is diagnosed. So in fact, in my clinic yesterday was a very interesting story of a child with a limb malformation. And the mum told me that when they did the scan at 12 weeks, there were 2 sacs there and there was a fetus. One was much smaller than the other one that had died. It didn't have a heartbeat. And so a limb malformation, again, is one of the malformations that's associated with twinning. It's a higher incidence in twins. What I wanted was a test that would tell me if there'd been a twin earlier that was either possibly detected or not suspected or detected, but was actually there. And I never thought that would happen. But then some very clever researchers in twinning from the Netherlands came up with, I'm not going to try and explain this in detail, but they came up with a test that you can do on an individual at any age that will reveal a specific chemical signature on the DNA that is stable and that is present in identical twins, but is not present in non-identical twins or in singletons. And I thought, well, this is our chance. So I started this project. Well, I spoke about it in, was it November 23? I had a US collaborator on board, but for various reasons, and some of you have provided samples for which I'm deeply grateful. The progress on that, and it's the laboratory side, because I did collect a good few samples actually, but for whatever reason, and sincere apologies for this, but beyond my control, that has not happened. So I'm now in the process of applying for my own funding in collaboration with this Dutch researcher who I really rate. And she invited me to a conference in Amsterdam last month, which I attended online actually. Wonderful conference where all people interested in twinning got together, and I gave my talk about this, which was well received. And we are now putting in a grant to work together on this, which I'm really excited about. So what we will be doing, it's a research study. We want a sample from the affected child or adult, OA, TOF, VACTERL, and some of you have done this already, a short clinical questionnaire. You have to sign a consent form. We can collect a swab from the inside of the cheek and extract DNA from that and test it for this chemical signature. It's not an individual feedback study at this point, and that's for lots of reasons, but it's to do with the fact that it's more of a statistical thing. So if you take a large number of identical twins and a large number of singletons, you can see a population difference. But if you look at a single individual, it's sometimes not so clear. But I would love to see this developed and potentially, if this hypothesis is correct, brought into the NHS, but that's a long way down the line. We've got a lot of work to do before then. So, thanks for your attention. Sue Lewis-Jones, all the TOFS committee, and if you've got questions, happy to answer. Thank you very much.
Ja, I'm listening.